So thank you all. And yeah, so the issue of genetically engineered trees does not only involve the American chestnut. As you mentioned, there were a couple other species that have been worked on over the years. The impetus behind genetically engineering trees came out of the timber industry. They were looking for superior quality trees for their plantations, for timber, for paper, easier pulping, et cetera, et cetera. They wanted trees that would be more profitable and easier to manage. So that was the real kind of beginning of the push to genetically engineer trees, and it was largely the plantation species that got the attention. So eucalyptus is probably the most genetically engineered tree in the world. Pine would be number 2. Poplar is right up there. Sweet gum, and there's a few others. The American chestnut was a tree that came along a bit later because the immediate reaction from the public to industry when they started talking about genetically engineering trees was very negative. And so they realized quite quickly they were going to have a public relations problem there. And so they were looking for what they called a test tree or a poster tree. That's what those were their terms. Anyway, they decided that the American chestnut was the tree that they were going to focus on. So the collaboration that worked on the American chestnut included a number of industries that were looking for the American chestnut to do things like reclaim strip mine lands to produce biomass, ethanol specifically, because the per acre amount of sugar produced by chestnut groves or chestnut plantations is higher, they were guessing, than corn plantations. So anyway, so they wanted chestnuts for a lot of reasons, but mainly for the public relations. The idea that biotechnology could be used to bring back a species on the brink of extinction. That's how they put it. There are, still millions of American chestnuts growing in the forests, and there are efforts by organizations like the American Chestnut Cooperators Foundation that are breeding wild American chestnuts, which Lois can talk more about, to become more blight resistant. And I think the ACCF is pretty clear that they're not 100% blight resistant yet, but that they are working with that goal in mind. Do you want to say a couple words about that?
Yeah, well, the ACCF says that if a tree can live for 25 years, that's a good indication that it's going to be blight tolerant. So blight tolerant, they'll get the blight, but it'll be blight tolerant, so it's not going to kill the tree, and that's the whole idea. So they've been doing this since, I think the TACF and ACCF started really about the same time, which was started in the 60s, early 60s, and then in the 70s. So it's been, they've had trees in the ground now since the 1970s, and we've been to a couple of their orchards down in Blacksburg, Virginia, and I've never seen such beautiful trees. The leaves are just deep green.
Now you're talking about conventional breeding.
Conventional breeding, exactly. They're taking crossbreeding 2 American chestnut trees, and they also are doing grafting. But it's easy enough to take pollen from one tree that's a really good tree and take it, wherever, and put pollen on another tree. So we do a lot of self-pollinating and stuff. But they've, just recently, they gave us 160 seeds to plant at New England Botanic Garden, and they feel that their trees are now ready to be put out. Not that they're, not every single tree is going to live for 20 years, but most of them probably will. So, it's just a matter of it takes time. If you have to wait, trees will produce in like 7 or 8 years. So, you have that aspect of it, but still it's a long process. A tree is a slow grower. So whether you do genetically engineered trees or pure American trees, it's a time thing. It's going to take the same amount of time. And it's just, to me, there's no reason to do genetically engineered trees, except for the people who are going to make money off of it, like the timber industry, like Ann said, people like that. But for us, people who just want chestnut trees in our yard, there is no reason to do genetically engineered chestnut trees. We don't need to do that. They have pure American chestnut trees that will do just fine. So why do genetically engineered?
Because I mean, maybe they produce in 7 or 8 years and you start getting nuts.
Right.
And you can plant some of them. So if they
Right.
die in 30 years, then you can get replacements. But don't we want great big,
Well, yeah,
beautiful-
but right. But I mean, if the tree is living for 30 years, most likely it's going to keep. It's not going to all of a sudden die of the blight at 30 years old. The blight will, if a tree's going to get blighted, it's probably going to happen in the first couple of years. You'll see blight at the root of the tree when they're really small. But I would say even if the tree lives 10 years, to me that's a good indication that that tree is going to be around for a long time.
Okay.
So and one of we'll get into some of the science of the American chestnut as well. And hopefully the wind won't keep blowing my notes around. But I wanted to talk a little bit more about the importance of GE trees and the history of GE trees for the eastern United States in particular, before we get totally into the American chestnut. But the story of the American chestnut is one that is a cautionary tale when it comes to genetically engineered trees, because of all of the hype about it being the Darling 58, which was the genetically engineered tree event, as they call it, that they were going with, that the American Chestnut Foundation decided to go with, that the SUNY School of Environmental Science and Forestry decided to go with, was this Darling 58 tree, and it ended up not working. We'll get into the details of that, but they applied petitioned the government for deregulation in 2020, when the trees were still 3 years old, arguing that the genetic transformation that they had made in the tree was going to be the solution. And at 3 years old, these trees had been in the field, outside in the field for 3 years. They decided that that was proof that these trees would be blight resistant. And 4 years later, they were suddenly 3 years later, actually, they were suddenly showing all kinds of problems, which they described as, the American Chestnut Foundation on their website describes as extreme variability in blight resistance, growth penalties upwards of 25%, reduced growth in trees carrying the transgene, the OXO gene, higher mortality rates in genes carrying the OXO gene, lethality in the homozygous state, that in other words, if an offspring inherits the transgene from both parents, it dies, and genetic deficiencies. So this was found 7 years after they started doing this collaboration, ESF and TACF. They found all of these problems 3 years after they asked the government for permission to release them into the environment. So had they been successful in getting their rapid permission from the government, which all of the hype from the researchers in the various articles where they were quoted, all of the newspaper articles and interviews that they did, they thought they were going to get this permission from the USDA Animal Plant Health Inspection Service like that. And because there was such an outcry from the public, they did not. And the USDA, I think, took a lot of time with this, not just because of public opposition, but also because they had never done this before. And that was something that was pointed out by the National Academy of Sciences in a report that they did on the potential for biotechnology in forest health situations, to improve forest health situations. And what they found through their study was that none of the agencies involved, the APHIS, USDA APHIS, the FDA, or the EPA, had any experience dealing with trees in a forest setting. And they had no idea what they were doing. And the researchers reflected that as well. In one of their webinars, they talked about how part of the issue with the process going slow is because the regulators really didn't know what they were doing. So it's been quite an interesting thing to watch and be involved with. On our side of it, we've been campaigning to stop the release of the genetically engineered American chestnut tree. And from our perspective, it was because there are just too many unknowns. So genetically engineered trees have never been done before in a way that they're released into the environment. In fact, they would be, if this petition was approved, they would be the first GE trees released into the environment with the intention of contaminating, spreading their transgenes to wild populations to reproduce infinitely. That's never been done before with any plant intentionally. There's been some escapes, but it hasn't been done intentionally. And so, this was something that was really a precedent-setting thing, had never been done before. We work with a lot of geneticists and other scientists who were very concerned because there was no documentation, there was no science to say what the impacts were going to be. These trees were too young, to have even a guess of how these trees would act in a forest ecosystem setting. You would have to have a few life cycles of the tree to see what happens to the tree through these life cycles, to the tree, to its progeny, and so on, as the gene is passed on to future generations. And of course, they didn't do that. They just created a tree in the lab. They put it in the field. They said 3 years later, it's still blight tolerant, so we're good to go. And 3 years later, they were all dying. So for us, it's a real cautionary tale, and it reminds us that the researchers can say they understand the science, they can say they have the science, they can say that things are safe, but the fact of the matter is with trees that can live, decades, if not centuries, there's just no way to know how they're going to respond in a wild forest setting with all of the different biodiversity, all of the different ways that trees interact in the forest ecosystem with each other, with the soils, with the microorganisms, with the other wildlife, with the other understory plants, et cetera, et cetera. The forests are the most biodiverse ecosystems, and so understanding how these GE trees that have never existed before in nature and evolution will interact with that forest ecosystem without doing long-term testing is impossible to know. And some of the scientists that we work with argue that you can never know it, that those risks can never be understood.
Can I say something?
Yeah, please.
Just genetic engineering is really a new technique. I mean, genetic engineering has not been around for 100 years, so it's a fairly new technique. And when you think about the life of a chestnut tree, it can be 500 years. So, and the chestnut tree interacts with other trees in the forest. I mean, they have a great root system, and that mycorrhiza stuff connects with other things under the. So there's all of these things that we don't really understand. So how can they say, yeah, this is going to be okay in 100 years when they can't even make him work for 3 years? It just doesn't make any sense to do that when it's not necessary. I mean, that's the whole. It's why do it? It's why take the unnecessary risk? What happens if something goes wrong 100 years from now? And there's chestnut trees, and chestnut trees will produce on their own, and they spread. What happens if these trees spread and something goes wrong? And it affects other things in the forest.
And one of the things that the American Chestnut Foundation has said since they released a press release in December saying that they were no longer backing the D58, the Darling 58 GE chestnut tree, one of the things they said about it was that even though it failed, even though it died, many of them died or had growth problems, et cetera, they were still safe. They didn't believe that these trees weren't safe. And based on the science that was in the petition, they said, we see that this is a safe tree. So even if it had gotten out into the environment, it wouldn't have been a problem. But the fact of the matter is these trees were meant to spread. That was their whole intent, was to cross with wild American chestnuts and spread their traits. So if these trees had defective traits that were causing, very high rates of mortality or other growth defects, and those traits spread to the wild population, I don't see personally how they can say that they would be safe, that there would be no problems there. They also said there were extensive safety tests during the petition that they were drawing on, saying, these clearly were safe. But if you actually look at the petition, and we have, some of the scientific analysis that I can share with anybody that's interested, they didn't actually use D58 material when they did their safety tests with amphibians, with bumblebees, et cetera, because they didn't have enough material. So with the bumblebees, for example, they were doing pollen feeding studies. They didn't have pollen from the D58s because they were too little. So they used pollen from non-transgenic American chestnut trees that they mixed with purified OXO, which is the gene that they put into the the enzyme of the gene, I'm sorry, mixing up my words. Anyway, OXO is what they engineered into the trees to make them blight tolerant. They took purified OXO from barley and they mixed it together with non-transgenic American chestnut pollen and then fed that to bumblebees and said it was equivalent, which it's not. Scientifically, there's no equivalence there. Bridging is what they call it, and that's clearly not what that is. With the case of feeding them to salamanders, they used D4, which was an earlier transgenic event that they had that had far lower expression of OXO in the leaves. They fed those to the salamanders and the other amphibians and said there was no problem, so therefore the D58 is not a problem, even though they didn't actually use D58 material. So there were serious problems with the petition from the beginning, even before all of these problems arose with the D58. And the little cherry on the sundae with the whole D58 scenario is that it turns out this 7 years of testing that the American Chestnut Foundation had done in collaboration with the School of Environmental Science and Forestry were on the wrong tree. So all of the public relations was around the D58, and all of the pronouncements were around the D58. The petition was on the D58, but in fact, there had been a mix-up right in the very beginning, and ESF had given the American Chestnut Foundation D54 pollen. So all of the experiments that the American Chestnut Foundation had been doing for those 7 years was on the wrong GE tree. So people have gone to the American Chestnut Foundation and said, well, if it's the wrong tree, why not go back and try the D58? And it turns out that it wouldn't have mattered if it was the D58 or the D54 because the constituent promoter, sorry for the technical jargon, the way that the transgene worked in the American chestnut was it was on all the time. So the blight resistance trait was on all the time, whether or not the tree was actually being attacked by the blight. And what that did, it had a metabolic cost, which ended up causing the trees to have these growth problems. They were putting so much energy into being blight tolerant in every single cell of their tree, in every single cell of their being, that they had growth problems, they had metabolic problems, and so on. So the American Chestnut Foundation decided that it didn't matter if it was D54 or D58, it was not worth pursuing. Besides which, as they pointed out, that the mix-up had happened so long ago, there may only be one D58 tree in existence anyway. So they just felt it was a lost cause and something not worth pursuing. So they pulled their support for the petition to APHIS. And ESF, however, is still is still asking APHIS for permission to deregulate these trees and still plans to move forward with them and with distribution, et cetera. I don't know how they're going to do that because they don't actually have these D58 trees, but they are trying.
Could you say what the impetus for that is? I don't quite get it.
The impetus for
continuing to promote defective trees.
Yeah, TACF in their website was trying to figure that out as well, and I think it might, my personal opinion, I can only give you my personal opinion because I'm not with ESF, is that the founder of this program, Dr. William Powell, who had put his whole life's legacy into the D58, he passed away last year before all of these problems were found, and so it seems to me that ESF is pursuing this in honor of Dr. Powell. They want to continue with his legacy and see if they can figure out a way to make it work. And TACF has said this isn't going to work. We can't. And TACF has also been very clear that they're not going to push this anymore or promote this anymore because they're afraid of the damage it will do to public relations. In other words, they're afraid, and this is in their literature, they're afraid of the impact this will have on people's trust in the genetic engineering of trees. Yeah.